Hi, so today I'm talking about how ATP is generated, and the first thing I'm going to talk about is glycolysis. So in step one, glycolysis occurs when glucose enters the cell. Here we have the cyclic glucose molecule, and it's going to go through a reaction that is catalyzed by a hexokinase. And it's also going to be a reaction that requires energy. It's going to consume ATP, and this glucose is phosphorylated into glucose 6-phosphate. Obviously, right here it's more polar, therefore it's destabilized, more reactive, more likely to continue on in glycolysis. It's also going to produce ADP. Step two, we're going to go over here. That same glucose 6-phosphate is going to be catalyzed by phosphoglucose isomerase. It's going to create fructose 6-phosphate with 6 carbon atoms. So that's something to note. We take that fructose 6-phosphate down into the next step. It's phosphorylated, consumes ATP again. Reaction also requires energy and catalyzed by phosphofructokinase. We're going to create 1,6-biphosphate, and then from there we're going to go and we're going to have another reaction catalyzed by aldolase. We're going to break down that fructose into two different molecules. We're going to have GAP, which is ready to continue on in the cycle, and we're also going to have DHAP. And each of these have 3 carbons in them. That's why we made the fructose. DHAP goes through another reaction catalyzed by triphosphate isomerase to create GAP. Now that we have 2 GAPs, it can continue on in glycolysis. We take the GAP, we have NAD+ in its oxidized form. It's going to be able to transfer electrons into this reduced form, NADH, 2 of those, as well as hydrogen atoms, and 1,3-BPG. So 1,3-BPG has a high phosphoryl transfer potential. That means it's going to be able to turn ADP into ATP, which is the next step. We have step 7. We take 1,3-BPG, we transfer ADP and hydrogen, we create 2 more ATP. It's catalyzed by phosphoglycerate kinase, and we create 3-phosphoglycerate. After that, we take that 3-phosphoglycerate, we have phosphoglycerate mutase, and that's the enzyme for this reaction. And then we're going to move the phosphoryl group from carbon 3 to carbon 2 over here. So we're going to be able to transfer that phosphoryl group. After transferring, we have 2-phosphoglycerate, and we're going to have a dehydration reaction. We're going to get rid of some water. You can see over here it's dehydrated, and we're going to also have PEP. PEP is going to go into our last step. It's going to be catalyzed by pyruvate kinase to give us our pyruvate in a ketone form where it's more stable, as well as 2 ATPs. So by the end of just glycolysis alone, we've produced a total of 4 ATP, but we've also consumed 2 ATP in the process. We've produced 2 NADH here, but each NADH is equal to, 1.5 ATM or ATP. So we're going to multiply the ATP. That gives us a total of 3, and we also got 2 pyruvate molecules that we should note because it's going to continue into our next step. So in the next kind of cycle or mechanism that we're going to look at, we're going to look at those 2 pyruvates that we have, and we're going to see how they turn into acetyl-CoA. So this is the step in between glycolysis and the TCA cycle that we'll look at afterwards. So this entire reaction is catalyzed by pyruvate dehydrogenase complex, which is made up of 3 enzymes: pyruvate dehydrogenase, dihydrolipol transacetylase, and dihydrolipol dehydrogenase. So in the first step, we're going to take a TPP right here, which is a coenzyme. And anytime we have a coenzyme in a reaction, we're going to need to regenerate it later. So that's just something we should remember. So we have TPP, and then we're going to take the pyruvate. We'll have 2 of these. Pyruvate, we're going to remove this CO2. We can see it's over here as a product. It's catalyzed by pyruvate dehydrogenase, and we're going to come up with hydroxyethyl TPP complex. We're going to combine those, kick off that CO2. Step 2, we're gonna have that complex, same complex, and we're gonna have another coenzyme, lipoamide, and we're going to then have a pyruvate dehydrogenase enzyme. So together they're gonna work, catalyze that reaction, and we're going to create TPP. Like I said, it's regenerated. We'll have to again regenerate this one in just a couple steps, and then we'll have an acetyl lipoamide with this acetyl group now bound to it. And then in step 3, we have coenzyme A and acetyl lipoamide, and we're going to have them react with dihydrolipoyl transacetylase, and it's going to give us acetyl-CoA, which is going to be continued on in the TCA cycle, and then dihydrolipoamide. So from here, we're going to be able to get the coenzyme regenerated so that we can end the reaction, and we take the dihydrolipoamide, FAD, which is going to oxidize this. The 2 hydrogens here are going to be removed. We're going to create a FADH2, and we're also going to regenerate the lipoamide. We're going to have those bonds reformed. So from there, we're going to use NAD, use that to transfer those electrons to create NADH, and we're going to have 2 of those because we had 2 pyruvate. So again, for this reaction, the total ATP produced would be 2 NADH, which are worth 2.5 ATP each. So we're going to have a total of 5 ATP for the pyruvate to acetyl CoA. So the last one I want to talk about is the biggest doozy of them all, and it's the citric acid cycle. So just to jump into it, we're going to start off with oxaloacetate and the acetyl CoA that we had produced, and we're going to go through a hydrolysis reaction where we're going to add the H2O. It's catalyzed by citrate synthase, and we're going to create citrate and CoA. So from there, we'll take the citrate catalyzed by aconitase, and it's going to be dehydration reaction. Let's get rid of that hydroxyl, and we're going to create an intermediate cis-aconitate and a water, because it was a dehydration reaction. That water is going to be used again in another reaction, same catalyst. It's a hydration reaction, and now we're going to add the hydroxyl to form acetocitrate. So in the next step, we'll take that same molecule, we'll use NAD in its oxidized form. It's going to transfer those electrons. We know we're going to get NADH, and we also got the oxidized oxalosuccinate. From there, we're going to have this reduced carboxylation reaction, and we're going to kick out that CO2, and we're going to create alpha-ketoglutarate. After that, we're going to go take alpha-ketoglutarate. We're going to jump down here again, use another NAD plus and the CoA, and we're going to have it catalyzed by alpha-ketoglutarate dehydrogenase. It's going to be the second, we just had that first one, it's going to be the second decarboxylation reaction, and we're going to take out another CO2, and we're going to have that right here, as well as producing NADH and succinyl-CoA. So in the 5th step, what we're going to do is take the succinyl-CoA. We're going to have this phosphoryl group, and we're going to have GDP. So in order for these to bind, it needs some type of energy for this reaction. When we break this CoA, this bond, we're actually going to generate enough energy for those to bind to give us GDP, and we know GDP later produces ATP. We also have the CoA and succinate. And if we do one of these, we go up here, we're going to have the succinate. We're going to have this oxidative form of FAD. It's catalyzed by succinate dehydrogenase, their whole reaction. And we're going to go to fumarate. And we're going to also, using these 2 hydrogens, create FADH2. And that's going to be in the reduced form. And then we're going to take fumarate. Here in step 7, it's going to be a hydration reaction. Add water, catalyzed by fumarase. We're going to add the hydroxyl to create malate. We're going to also create NADA+, that NADA+ and malate down here. It's going to be catalyzed by malate dehydrogenase, and we're going to end up with oxaloacetate. and that's it in its oxidized form as well as NADH. So at the end of all of those reactions for just the citric acid cycle, we had produced 2 GTP, which is the equivalent of 2 ATP, 3 NADHs, each are worth 2 ATP, or 3 NADH, and we did 2 of those because we started off with 2 acetyl-CoAs, and then we had 6 total NADH, each worth 2.5 ATP per NADH for a total of 15 ATP. Same goes for the production of 1 FADH2. We had 2 of those, we got 2 total, multiplied by 1.5 ATP each to a total of 3 ATP. So that, super easy, is how we get 30 ATP generated for 1 glucose molecule.